Thanks for having me. I'm between you and the next break, so I'll drop some slides out. So I want to give you a little bit of overview of what I do here and then tell you about the subset of topics that I'll talk about today. So my group does a variety of things. I'm actually a microbiologist by training, and a lot of my grad work was on making microfluidic and lab-on-a-chip systems. So I've always been interested in this interface between nanotechnology and biology. And I continue to do that work here. I've been here about 13 years running a research group. And so one of the ways we look at this interface is through using nanotech to do a better job of understanding biology. And that's where we work on systems like microfluidic platforms or lab-on-a-chip systems where we can manipulate these structures on a small scale and then use them for biological studies. We've done a lot of work with cell printing and patterning, and I'll show you a little bit of work on that topic. We do biosensor research. That's the last set of slides I'll show you is about some of the work we're doing on a new sensor for Lyme disease, which is a tick-borne disease that we have here in the Northeast. We also use nanotechnologies for more applied biology. And so we do things like make scaffolds and structures to support tissue engineering. So if you're growing cells on a pattern substrate, for instance. I'll show you an example of where we did work on this to structure surfaces to limit the ability of bacterial cells to attach to that surface, which is actually also of interest if you're looking at mammalian cell systems where you want to make a pattern surface where you can then encourage cells to attach or grow in a certain manner. I won't talk today about work that we're doing in an area that I tend to call biomimicry. It's really my excuse for saying we're doing electronics, but it's somewhat related to biology. And so we do a lot of work more recently on neuromorphic computing, but especially developing unique nanoelectronic devices to mimic the synapse in the brain. And so I'm sorry, I won't give slides on that today. Just didn't have enough time to put all these topics into one slide deck. So the first thing I'll tell you about is work that we did specifically with atomic force microscopy to measure the mechanical properties of bacterial biofilms. So I figured since Park was inviting me to talk, I better talk about AFM. So for bacterial biofilms, first of all, has anybody heard of a biofilm before? A few of you. Okay, if you run your tongue over the top of your teeth right now, you will be interacting with a biofilm, and that's the plaque on your teeth. So plaque is actually really gross. It's about many hundred species of bacteria that form this layer. And it's not only the cells themselves that form this layer, but all of the gunk that they produce. And we tend to call that extracellular matrix type material or extracellular polymeric substances. And this is all the goo that those bacteria produce to hold them together and also attach them to the surface. And it also limits the ability of things like antibiotics or your mouthwash to penetrate into the lower layers and kill off all of those bacteria. So what's kind of interesting about this is that bacteria have this whole life cycle. So the ones that do form biofilms, they're usually free swimming. They call this planktonic, like plankton in the ocean. They can then interact with the surface and attach. They can start to grow. They start making this slimy material we call EPS. They then can grow into really large structures. They almost look like they're vegetative, like a mold or a slime. And then eventually they're actually giving off other cells to start new populations elsewhere. So it's useful for things like biofuel cells, where you might want organisms to grow and do things, or even in sewage treatment systems, where you need these bacterial biofilms to do the work of that system. But for areas like in our mouth, where we have plaque, or any other area where we may have biofouling, we actually want to prevent that formation of this biofilm. And so we can study biofilms in lots of different ways. And just to cut to the chase, the things that we were interested in measuring when we were doing this work was how do things like the environmental conditions affect how those organisms are able to attach and how that biofilm acts and how we might be able to mitigate its formation. And specifically, we ended up focusing on things like fluid flow and shear, and then also what are the mechanical properties of those films while they're in place. So we used a combination of confocal microscopy. Probably most of you are somewhat familiar with confocal microscopy. It's essentially an optical imaging approach where you take thin slice images through a sample in a Z-stack and then recreate a 3D image. We specifically use fluorescence confocal. This is a laser scanning fluorescence confocal microscopy. So what it requires is that you have to label your sample with a fluorophore, and you can label different regions of your sample with different fluorophores. If it's a biological sample, you can tag things with things like antibodies, and then sort out those different features afterwards. Unfortunately, this wasn't a Park system, but we were originally working with a Bruker system, their bioscope, to do AFM in conjunction with confocal. And I know that's kind of somewhat standard these days, but when we first were starting this work, this was novel. So we purchased a Leica SP5 confocal microscope, which had a Bruker bioscope catalyst mounted on top of it. And so we could actually swap the stage and go into regular confocal mode with a normal confocal stage, or take that stage off and put the AFM stage on that and use the AFM head in conjunction. So I don't have to tell you about force spectroscopy curves. For measuring these biological samples, and I don't have a slide on this, we actually are using, when we're looking at bacterial biofilms, that material is really low modulus. So we're talking in the kilopascal regime. So it's gooey, and it's actually really a viscoelastic material. So your normal sharpened probe just doesn't cut it for trying to make mechanical measurements of that material. So this picture is deceiving. We actually were mainly using 2 to 5 micron spheres attached to the probe tips, and using that sphere-mounted cantilever as our measurement probe. So what we needed to do, though, is to create a system where we could establish that biofilm in a fluidic system, and then kind of like a convertible, pull the top off, and then gain access with the AFM to be able to image with confocal from the bottom, and then image with microscopy from the top. So that's what this is showing. So this is a large format slide, so they are approximately 2 by 3 inches. These are microfluidic channels. This is actually multiple layers of silicone, molded silicone, that create this structure with all our input and output tubing. And from the cross section, it looks like this. We do inverted confocal microscopy. So we image up from the bottom. This is the fluid chamber. We grow our cells or our bacteria by flowing nutrients through here and establishing that biofilm. And we can pop this layer off, this convertible lid, and then move in with the atomic force microscope and start making measurements. So this is an example showing, this is Pseudomonas Aeruginosa, which is a really commonly studied organism that forms biofilms. And I think if I click this is what it looks like in real time with a confocal. These cells are moving across the surface, partially because it's under flow. They start to attach here, and they proliferate. And eventually they're moving out of the image plane and blurring here because they're actually growing up on the surface. This is just bright field taken with that same confocal. So now we've established that we can grow these biofilms in this microfluidic. We can go around and start poking it with the AFM and measuring the modulus of different sections of the film. So this, I always think it looks like an island I'd like to be on in the winter in Albany, which you guys are experiencing today with the snow. But we went around to sections of these biofilms and probed, basically took force curves at these different regions. And I don't show any detail here, but we had to then extrapolate those data out. At the time we were doing this work, there was no good software in the Bruker package to be able to extract the modulus information that we needed for these curves. So we typically took force distance curves and then applied like a Hertz model to them, knowing the size of the indenter, which was the sphere mounted on that tip, and we were able to extrapolate out the modulus of the material. So these are all showing, and we were kind of in the 1 to 2 kilopascal range, pretty much regardless of where we were in that biofilm. And again, I'm not showing the data here, but this correlates really well with measurements like rheometry, where you can do bulk measurements of a biofilm and show that these are typically in this really low modulus regime. What we weren't able to do, and we didn't really pursue too much, was to try to extract out viscoelastic properties. And so this is something that is actually probably a more interesting way to try to study these films, because they really are so fluid-filled and act really like a hydrogel material that even just this regular elastic modulus measurement doesn't really do justice to what's going on in the rest of the film. We've continued some of this work. We've looked at other biological tissues like mammalian tissues that are growing. So we work with a group that does salivary gland development work, and so we were able to build microfluidic devices that could hold on to pieces of tissue, and while we were able to make measurements with AFM. And then we have a number of groups here that have done similar measurements where they do things like create a scaffold type of material and then grow cells on top of it and try to make combined mechanical measurements of both the scaffold and of the biological tissue growing on top of that. So kind of a neat platform technology to be able to do this combined 2 types of imaging and measurement. So I mentioned that we also do work on surface topography and how that affects how cells interact with surfaces. And so some of the inspiration for this work came from this interest in biomimicry. It kind of came about when people were first talking about nanotechnology. They said, hey, this gecko foot model, right? How does a gecko walk up a wall and stick to a ceiling? Or how does the lotus effect work? So when you see these plants that create these really small micro and then nanoscale nodules on their surfaces, and water. It actually acts like a super hydrophobic material, so water droplets bead right up on it. Similar to rose petals that also cause this water beading type of effect. But in these two cases, it's very different. This structure is, it causes these droplets to just roll off the surface, whereas on a rose petal, you actually get pinning of the structure to the surface. And so all that comes into the actual morphology of the material, and also some of these really interesting Cassie-Baxter wetting type of phenomena that go on at that interface. Here's the Gecko foot model that allows for adhesion through van der Waals forces to structures. And then this is kind of neat. This is a Sharkskin. Some of you may have heard of Sharklet. This was a commercial product, and people were making swimsuits. I think it made it into the Olympics even, with this patterned surface to reduce drag while swimmers were swimming. So we said, like some other groups, this would be really interesting to try to mimic some of these surfaces and see the effect on cell attachment. And so again, we weren't the first to recognize this, and people have recognized there were lots of parameters from the surface structure itself to the surface chemistry, the wetting properties of the surface, and how that actually affects attachment. So I'll go through these quickly. We've developed several platforms to study this. So one was a microfluidic platform where we took a textured surface, and typically what we do is we generate that textured surface using one of our silicon micromachining techniques. So either MEMS scale device fabrication, or we also use structures that we build. I don't know if you get a tour while you're here on site, but some of the 300 millimeter wafer processing facilities where we're processing down to, tens of nanometers in feature size. So we'll take that type of master mold, and then we'll typically mold something like a silicone polymer or hot emboss into other polymers to generate a replica of that surface so we can reuse the mold many times. So we typically combine that with a cover that makes this microfluidic environment, and then we're able to ask questions like, how many E. coli bacteria stick to this surface if you just have no flow, or if you have fluid flow with this thing right side up or upside down? And we can look for binding to all these different types of structures. And what we saw with these early studies was that it was really kind of interesting. Bacteria tended to stick to features that were linear or protruding up out of the surface, but when you gave them a lot of divots or little holes, you reduced the ability of cells to attach to those surfaces. And so all of these lower cell densities corresponded to these kind of high density hole structures. So kind of interesting. So the next thing we did was say, okay, we've got these somewhat micron scale structures that we've been working with. Can we start looking at other more complex structures down into pushing into the nanoscale regime? So as I mentioned, we were able to use the wafer processing facilities here to structure all kinds of different checkerboard patterns. One of these looks like parquet flooring here. This is a mimic of one of these commercial products, the Sharklet surface, just on a smaller scale. And then we ask questions like, if we expose this to bacteria, do they stick? And then if we keep giving those bacteria food, do they form biofilms once they've stuck? And so this slide shows a couple of things going on at once. So each one of these subpanels is one of these areas of topography. And what we did to make it a little more interesting was everywhere, so this is a bunch of essentially little pillars sticking up. So they're pillars on this side, and then the equivalent size of space, so where the pillar would be, the lateral spacing is the same, but instead of a pillar here, there's a hole that's the same exact shape as the pillar. So raised features and recessed features. And so that's the same for all these, just with different types of patterns and different types of spacings. The red spots are where we image cells attaching to begin with. So we put flowed bacterial cells across the surface, and we look for where they stuck to begin with. And interestingly, we got a lot of binding to these recessed features, and then on some surfaces, they just stuck a lot to begin with. And as I mentioned, we gave the bacteria some food and let them go for 24 hours. Is it 12 or 24? It's been, 24. And then could see where these biofilms would grow. And not surprisingly, the biofilm tended to grow where the cells were sticking to begin with. They also form bridges sometimes, so if they stuck here really well, they could bridge across the areas where they weren't necessarily sticking much to begin with. But we noticed this trend of stuff sticking where we had these raised features, but not as much where we had the recessed features. So it was consistent with our earlier studies with E. coli that if we form these really small recessed structures that are closely packed together, we essentially limit the amount of surface area those bacteria are able to grab onto. We may even have effects of creating air pockets within those structures as well, because if you think this is all in silicone, so it's a very hydrophobic surface. And so We haven't gone on to study this in too much more detail about all of the phenomenological events that are going on to cause the attachment, but we're able to use this as a kind of a testing or screening platform to see what different cell types do on a variety of different surface topography. Okay, so one of the tools we work with is we do bioprinting. This company, Bioforce, I'm not even sure if they're in business anymore, but they used us as a beta test site for their equipment quite a few years ago now. And it generated some interesting work in our group, but also we were able to build a new type of printing probe for this instrument to do direct cell printing. So what this instrument is, it looks kind of like an AFM, except that the cantilever has a groove in it and a little reservoir, and you essentially use it like a quill pen. So it's like loading up a little old school quill pen with ink, which fills this reservoir, and then this is actually inverted when it's printing, but that tip now makes contact with the surface and transfers a little bit of ink. So this is back when people were doing a lot of dip pen lithography, and this was the idea that instead of having to dip and print and then dip again, you could just fill a reservoir and keep printing quite a bit more. So we use this. We printed a bunch of different things on surfaces. Here's an example of printing fluorescent proteins in arrays. You can imagine all kinds of array type of experiments you could do with biology. We then said, well, this is really good for printing molecular scale, small diameter. So these spots are, in the few micron scale for spot size. We had a lot of projects where we wanted to say, could we print whole cells and make basically a living pattern surface? The problem was we couldn't fit cells down these narrow channels. So they were narrow, but they were also very shallow. So a cell just would not fit down these channels. So what we did was we came up with an approach to make a polymeric set of printing tips that were the same platform format to fit in the instrument, but had a much wider groove and actually an encapsulated channel. So this is a process that uses SU8 photoresist. It's a negative tone photoresist, which cures kind of like an epoxy. So you can make a base layer, you can outline the base of your probe, make a channel layer. This is the cross section, this is top down, and then you can actually put a lid on top of your channel. And so you make an enclosed printing tip so that fluid can go down that channel. We had problems initially with fluid kind of leaking all over the place if we didn't put this cap on the channel. And they look something like this. So here's this pretty big, actually, proboscis, I like to call it. It's almost like a bird beak that's coming out. And so we can fill this up with fluid and then have a little meniscus on the end and print fluid containing cells or other materials onto surfaces. And so just some examples of what we did with this. This is printing mammalian cells in a matrix material and allowing them to grow for several days. If you print cells out onto a surface and there's no means for those cells to attach, you can make a little droplet, but then the droplet goes away as soon as you cover it up with growth media. So what we developed, we actually developed two different methods. One was to print in an alginate matrix and then expose to a vapor phase of cross-linking agent to cross-link that matrix after printing. The other thing that we did was we worked with a cross-linkable matrix material, which is basically a gelatin-based material that you could cross-link with blue light after printing. And so that's what this is right here. And so we've printed that cell and these cells in a little blob and then let them grow. And then they would actually fill that blob. And eventually, if you let them go long enough, they'd start to escape. So an example of being able to print little cells into arrays like this. We also did work to kind of combine the two things. So we used the original probes in this system to draw lines and spots. This is on top of a microelectrode array. And so here we drew lines of proteins that would be able to capture cells after the fact. And this is an image showing these are where neurons, so this is neurons that were harvested from rat pups, were seeded onto the surface. They affixed themselves to the spots where we had printed this capture protein, and then they were able to grow together in all these neural processes. And later work that this group did was able to show that they could measure communication between these neurons. So kind of a cool combination of being able to print directly on a surface and then capture cells and use this as now a functional system. So last thing I'll tell you about before you have your break is work that we're doing on Lyme disease. And so just show of hands, is anybody familiar with Lyme disease? Okay, so some of you. Probably a lot of you raised your hands, live somewhere east of Ohio. So Lyme disease is really spread. What it is a tick-borne disease. So we happen to have quite a few of these ticks around here. They're nasty. Okay, they are these tiny little insects. They will bite you, and if they're carrying, they carry a lot of different diseases, but they can carry this bacteria called Borrelia Burgdorferi, which is a spirochete. Even the bacteria looks kind of scary. They're shaped like these corkscrews. Turns out syphilis and some Rocky Mountain spotted fever are all also members of this family of bacteria. And if you're lucky and you get bit by this bacteria and it transmits this, or get bit by this tick and it transmits this bacteria, you will end up with a bullseye rash, which literally looks like a bullseye, and that'll tell you you need to go to the doctor and get antibiotics. And usually your antibiotic treatment will work and you'll be okay if you can identify this rash early on. The problem is lots of people either don't see the rash, they don't develop a really distinctive rash, and they miss it. And they end up feeling, have poor health, there's all kinds of complications from this disease. They end up going to the doctor says, I don't know, you could have all kinds of things that cause you to just feel kind of logy and not well and achy. And so what the CDC did was to come up with a criteria for testing that's really quite complicated. To say, well, did you get bit by a tick? No. Well, we can start this whole profile of tests, which include things like immunofluorescence assays and Western blot analysis. The problem is none of these tests are very accurate, and they also take a lot of time, and they're very subject to interpretation. So when people look at the results of something called a Western blot, they're looking at a control set of these little lines, and this is the patient sample where they have to interpret, do the lines on the patient sample, do they match the control? Are they brighter than the control? Are they weaker than the control? And so it's very subjective to interpretation. So what we've been trying to do is to say, well, can we develop a better platform technology for doing this type of analysis? So we work with a company called Ciencia out of Connecticut. Turns out Connecticut is one of the states that's really bad for Lyme disease as well. And they developed a technology called grading-coupled surface plasmon resonance. And if you're not familiar with surface plasmon resonance, the basics of it are that if you impinge light on a metallic surface, you can excite an oscillating wave of electrons on the surface called an evanescent wave, or a surface plasmon. And if you change the local index around that surface, you can change the required angle of incidence to generate that surface plasmon. So it's measurable output, so when you impinge different angles of incidence, or if you change the index on this surface. So it makes a really sensitive technique to measure surface interactions. So they developed this instrument to be able to do this type of analysis, but in an arrayed format. So they actually look at it from the front. Instead of a prism, they build a grating on this surface, and you can look for all kinds of interactions all at once across that entire surface. We use it, we started this work by just building them these gratings. So we used our foundry here, our fab, to basically do photolithography and pattern lots and lots of replicate grating chips across a big 300 millimeter wafer. And in cross section, this is kind of neat. We developed a sinusoidal etch to give it kind of this sinusoidal pattern on the surface. This period is 500 nanometers between each of these ridges, and we're etching only about 50 nanometers into the wafer. We coat that with an adhesion layer and a little gold capping layer. You need a noble metal like gold, or you can also use silver to get a good plasmonic effect on these surfaces. And what was interesting about this is that not only can you do surface plasmon resonance, but you can do plasmon-enhanced fluorescence or other plasmon-enhanced type of spectroscopies or measurements. So in the case of what we're doing, we're actually not even using the normal SPR mode for these chips. We're simply using it as a fluorescence enhancement surface. So we shine light on the surface, it generates a surface plasmon, and if we have a fluorescently tagged molecule close to that surface plasmon, we actually generate about 100 to 1,000-fold more fluorescence intensity than if this was just on a piece of glass alone. What this shows here is that this is on one of the gratings where you generate a plasmon. These are spots where we have a fluorophore attached. Here's on a flat surface where we have basically see no fluorescence enhancement. So the grating matters, and this whole arrangement for doing this plasmonic enhancement matters. I'm going to skip a little bit. We do a lot of work to generate the biological side of the assay. So we express proteins from these organisms that we're interested in detecting, and we print them as spots on these chips. We do a lot of work to optimize how this works. What we've been able to show through a bunch of different studies is that we're getting down to the femtomole amount of target that we can capture and detect on that surface. So that's pretty darn good for an assay like this. We've done a lot of optimization too with the sample concentration. Most of the studies that you do for something like a Lyme disease diagnosis is from blood serum. So nobody likes to give a lot of blood, right? So what we're able to show with this work is that we typically use dilutions in this kind of range. So we can take about 3 to 4 microliters of blood. So that's less than a normal droplet of blood. And we can then use the serum from that amount of blood. We dilute it into a running buffer and then run that through our chip. And our total time to detection is about 30 minutes to go from beginning to end. So very small amount of blood and very fast detection time. I will jump to this slide, which I think is probably the most important that we've done so far. We've partnered with CDC, with something called an organization called the Lyme Disease Biobank, and then also locally here, our State Department of Health, Wadsworth Institute, to get human serum samples. So we've taken serum samples they've collected from people who either had Lyme disease, did not have Lyme disease, or had what we call early Lyme, where it's not really detectable with the regular detection methods, but hopefully it's detectable with some new technology that somebody comes out with. And so this is a really boring graph except for the yellow here. So essentially we replicate what you can do with regular testing. So when there's a positive, we detect a positive. When there's a negative, we detect a negative. What we thought was really encouraging though is that for a lot of these samples where they're early Lyme disease, we can detect a positive in a lot of cases when the normal testing routes, so immunofluorescence or Western blot analysis is detecting a negative or giving a negative result. So we have some evidence now that this very high sensitivity, high accuracy type of platform that we've been developing is actually able to detect Lyme disease in patients before you could detect it using normal methods. So we're kind of excited about this, and our next steps are to try to expand the sample pool and look at many more patients to make sure that we can back up these data. I'll leave you with one more thing. Again, these graphs are kind of hard to see and maybe a little bit boring, but the results are really kind of cool. If you get an infection, your body mounts an immune response and you have a lot of antibodies generated during that immune response. As you clear that disease and you're no longer sick, that antibody titer peaks and then starts to go down. So eventually you should have a tapering off of that concentration of antibodies in your blood. The problem with Lyme disease is that people have symptoms that last a long time, even after they've actually killed off all of the bacteria in their system. They feel terrible, but they don't actually have the disease. And it's controversial because they're actually getting treatments like long-term antibiotics, which are detrimental to the rest of the patient's health, but the patient feels sick still, even though they really don't have active disease. What we've been able to do with these data, and again, forget the graphs, listen to me. What we've been able to show is that we can detect, in acute phase, we can detect a high antibody titer, and that's all the dark gray bars in all of these. These are all proteins from the bacteria. All the dark gray bars are the high antibody titer we detect against those particular antigens. When we look at a patient who's cleared the disease or who has had antibiotic treatment and is about 90 days post-treatment, we can now detect those lower titers, and that's these lower gray bars. This is really important for doctors who want to be able to work with a patient and say, look, your body has actually stopped the response to the organism. Your antibody titers have gone back down. And now really what you are is in what they call a chronic Lyme phase, where really there's other things going on in your body. Some people are having like certain autoimmune type of responses or other effects from the disease that aren't necessarily active infection. So this is becoming kind of an important tool to be able to have to help doctors work with their patients with Lyme disease. So I'll leave it at that. This is my group here at SUNY Poly. And yeah, we've had funding from NIH, NSF, Bay Area Lyme, CDC, and a variety of different organizations. So thanks a lot for your attention. Yeah, so what we've been able to do is we can do comparisons. Some of this work was that the company did before we got involved in using their instrument. When we measure just a flat gold surface versus that corrugated or sinusoidal grading type of surface, we see between 100 and 1,000-fold enhancement in the fluorescence intensity. Our collaborators from Ciencia have done work where they actually looked at the distance between that grading surface and where the fluorophore sat by doing layer by layer deposition. And so what they actually find is close to the surface you have a quenching effect, and then at a key distance from the surface you start into enhancement, and then that peaks, I think, in the kind of 5 to 10 nanometer range from the surface before tapering off again. Yep.
So I had a question about the printing of fluorescent proteins.
Yep.
How far could you scale that down? Could you deposit single molecules?
So Yeah, you can do it by dilution. So if you're printing spots, so regardless of the spot size, if your printing solution is dilute enough, you have some probability of printing one protein at a time. And then in a combination between spot size and dilution factor, you can start to approach that. The problem we had, and we did this with not down to the molecule level, but down to the single cell level or the single, usually like nano or micro bead level, is that then it's kind of a statistical distribution of whether you print anything at all in each spot. And so that's where you're stuck after a while, is then confirming, did you actually put something in the spot with this combination of spot size and dilution factor. That's our challenge that we had with that approach. Yeah, so we could, and so it's easier with beads or cells. Bacteria were kind of fun. We printed bacteria, and you could go back and image and see what you ended up with in each one of those spots. But it's like a Poisson distribution of what you actually end up with. Yeah, you could. Yep. Yeah. Okay. Well, thank you.
Thank you.